Let's begin with a description and definition of histology. The cells in the body are organized into tissues, and together these tissues make up the organs of the body. The human body is made up of four main tissue types: epithelial, muscular, neural, and connective. Some tissues, such as smooth muscle for example, are in many locations within the body, whereas heart muscle tissue exists in only one location. Histology is the study of tissue, and histopathology is the study of diseases in certain tissues. In order to study tissues, sample preparation must be done. And this is because biological tissues are not strong enough or rigid enough to allow them to be cut in thin sections for study. In order to facilitate sectioning the sample, it must be embedded in something such as paraffin wax by placing it in a liquid wax and then allowing it to harden. However, prior to embedding the sample for sectioning, it must be preserved or fixed with a formaldehyde solution, then processed or dried with grated alcohols. The tissue samples themselves can then be stained or treated with labeled antibodies for microscopy. Examples of stains commonly used are hematoxylin and eosin, or H&E, gram staining for bacterial identification, and DAPI, which is a fluorescent stain to label DNA in the nucleus. The scope of histology. Histology is central to pathology, general medicine, and science itself. Anytime a sample is collected or a biopsy is taken, it typically must be subjected to histological examination to determine if there's any disease or change in the tissue. Histological examination of a biopsy plays a very key role in the diagnosis of cancer. For example, in this photomicrograph, it's evident of Kaposi's sarcoma, which is a cancer in the dermal layer of the skin.